Well, thank you for joining us on this sort of late time slot on a Monday evening. My name is Rachel Hill. I'm head of the Behavioural Neuroscience Lab, which is part of the Department of Psychiatry. And what we're trying to do in my lab is really try to understand the neurobiology of schizophrenia, which is a very severe psychiatric disorder that affects about 200,000 Australians. And it's characterised by very frightening hallucinations and delusions, social withdrawal and cognitive impairments. Well, current treatments for schizophrenia are antipsychotics, and they can treat some of the symptoms, some of the psychotic symptoms, but they come with quite nasty side effects, and really they're not doing a good job, essentially. So what we want to do is really better understand the neurobiology of the disorder so that we can design better treatments that actually modify the course of the disorder. The way we do this is to follow this pipeline of translational research. So we have clinicians within our group that identify and characterise individuals as well as their families with schizophrenia. We also collect serum samples from these patients and do whole exome sequencing to identify genetic variants that might increase risk for the disorder. And we use post-mortem tissue to try to substantiate and localise putative molecular and cellular changes that are altered by genetic variants. Post-mortem brain work obviously has its limitations. You can't get access to much post-mortem tissue. Also, it's more representative of the end stage of the disorder. And so to overcome these challenges with post-mortem tissue, we also use animal models. And here we can create transgenic animal models of the risk genes that are identified. And using these animals, we can probe behavioural consequences of risk genes. And also what we can do that we can't do in the human is to really look more closely at the brain and probe neurobiological effects of risk genes to try to identify new treatment targets or new biomarkers to then take back to the clinic again. So some examples of some projects that we currently have running in our lab that use this pipeline is a project which is looking at ARX, a hub gene for a common biological pathway in schizophrenia. So we identified a patient with schizophrenia that had a mutation in this gene called ARX, and we have created a mouse model with this mutation inserted into the mouse DNA using CRISPR-Cas9 technologies. And this project will be to probe the behavioural and the electrophysiological consequences of partial loss of ARX function, and to also probe biological pathways that are altered by ARX loss of function to try to identify a common pathway that's altered in schizophrenia and hopefully new treatment targets to bring back to the clinic. We also have a project that's understanding the role of a molecule called beta-cellulin in schizophrenia. So here we've actually identified from post-mortem tissue that a molecule called beta-cellulin is reduced in the brains of people with schizophrenia. It's also reduced in the blood. And to try to understand what this molecule does in the brain, we've created a transgenic animal model that's a knockout of beta-cellulin. And we're probing the behavioural consequences of loss of beta-cellulin in these mice, and also the neurobiological effects of loss of beta-cellulin. And we actually think that beta-cellulin might be a novel new treatment target, and we're trying to test this in these mice to hopefully take back to the clinic. And finally, a study that we currently have ongoing in the lab, and we have projects on offer for, is understanding the impact of COVID-19 exposure in utero on neurodevelopment. And this study is really born from past historical pandemic data that identified that infection during pregnancy is a risk factor for the offspring, so for the baby, to develop schizophrenia later in life. So we really wanted to try to understand how this risk factor causes neurodevelopmental delay. And so we model this in mice. So we inject pregnant dams with a viral mimetic. And then we look at the offspring. So we look at the behavior of the offspring and we look at their brain and different molecular pathways to really try to identify whether we can actually prevent the onset of neurodevelopmental delay and bring that back to the clinic. We also have a project on offer where we are prospectively monitoring children whose mother presented at Monash Health with COVID-19 infection during pregnancy. This is an ongoing project, and one of our students, Nihaan, is going to now talk to you a bit more about this project. So I'm going to leave my presentation. I'm going to introduce Nihaan, who's an honours student with us working on this project. Stop share. Take it away, Uni.
Thanks, Rachel. I'll just share my screen. Are those the presenter slides that are coming up, or the presentation?
Yep.
Yeah, that's good. Okay. So hi everyone, my name is Uni, and I'm just here to share a little bit about my experience as an honours student under the supervision of a Professor Hill and Professor Sanjam this year at the Department of Psychiatry. And as you can see, I've chosen a very topical project to get involved with. But before I talk more about that, I thought it might be helpful to briefly mention how I ended up as a student researcher on this project. So after the chaotic year that was 2020, I decided to take intermission after my 4th year of medicine and to try doing some research instead. And I can say without a doubt that I did not regret my decision one bit, and I would very highly recommend any prospective honours students, especially the med-sci students out there, to just take the plunge. Prior to this, I hadn't had much experience with research during my undergrad, but I knew I was drawn to academic psychiatry, translational medicine, and also the physician-scientist pathway. As a massive journalist at heart, I had a lot of trouble initially picking between doing a lab-based project, because on one hand, I sort of saw that as an exciting new challenge and as a way to gain a very valuable skill set I didn't have. But also at the same time, I knew I loved the clinical side of things and I didn't want to go a whole year without patient contact. However, I talked to Rachel about this, and in the end, I was very lucky because I actually ended up getting the best of both worlds with this project. So I advise anyone who might be stuck in a similar dilemma that it's definitely possible to look for a project where you can do both. So back to my project, a little bit of background. My research this question this year is, how does maternal exposure to the novel SARS-CoV-2 virus affect the neurodevelopmental trajectory of offspring? And the specific aim of my project is to investigate whether modifications to the epigenome may be a biological mechanism by which infection with SARS-CoV-2 may disrupt neurodevelopment. As tends to be when it comes to studying the brain or understanding how mental illness comes about, there's so much we don't understand, but from influenza epidemics to parasitic infections, for a long time now, there has been growing evidence to suggest that the infection during pregnancy may have a key role in the pathogenesis of neuropsychiatric disease. Our behavioral neuroscience lab have been using the maternal immune activation model to study in mice the various behavioral and cognitive deficits that maternal infection can confer to fetuses while they're in utero. With the ongoing pandemic and the novel SARS-CoV-2 virus and all its unknowns, suddenly it has become extremely relevant that we take a closer look at how environmental stresses like viral infections can affect vulnerable developing brains, and moreover, how this exposure in the womb may create a blueprint that may alter an individual's neurodevelopmental trajectory throughout their life. My project this year has taken place in the context of a much larger longitudinal birth cohort study, following up babies whose mums had COVID while they were pregnant. In order to draw out both the more immediate and contextual effects of stress from a pandemic to the possible long-term disruptions to brain development, the study design aims to follow our participants from birth to age 15 at key neurodevelopmental time points and collect various data sets at these time points to look at how these COVID-exposed children are growing and developing, and whether they might be at risk of neuropsychiatric illness such as ASD and ADHD. This year, I was able to help conduct our first follow-ups with mums and babies at 3 months, which was a massive highlight. The babies were so adorable. And speaking of babies, I did very much feel like an infant myself, starting out from the very basics in the lab, such as learning to pipette for the very first time. But now I've actually graduated to running some basic protocols independently, from learning to do DNA extractions from the buccal swabs we collected from our participants to use these downstream in analysis, to PCRs and other bread and butter techniques. Importantly, as a student, I've also gained some useful and transferable skills in terms of data analysis and working with statistics. In particular, this project has given me the opportunity to work with big data and dabble in bioinformatics, which I think is really incredible. And just to finish, before I came to this lab, I honestly didn't realize what a special environment it would be. It's been amazing to pick the brains of all the PhD students and the postdocs, attend lab meetings and listen to people present their research or scientific papers. And also, it really is special to be part of a team where scientists and clinicians are working so close together and where you can see that translational work from preclinical to clinical happening, and it's so rewarding. And overall, it's really been such a privilege having this opportunity, and I've learned so much from this very passionate and dedicated group of people. And I think with mental health coming into our collective consciousness as something that is more important than ever, I really, really think that the research we're doing in this group is so significant. And if you think so too, we'd love for you to come and join our team. Yeah, that's the end of my presentation.
Thanks so much, Uni. That was great. We're now going to hear from Witaya, who's a senior postdoc in our group. Witaya.
Can everyone hear me? Okay, I'm going to share the screen. Give me a second. Can you see my screen? Okay, my name is Witaya. I'm a postdoc in the group, and thank everyone for coming today. First of all, I would like to give a very quick overview of next generation sequencing. So it's a high throughput measurements of DNA and RNA to understand the genomic profiles and gene expression in different conditions. For example, in this case, it would be the healthy versus disease conditions. However, this method overlooks the fact that cells are functional units of tissue that has pretty unique gene expression and specific signature of epigenetics. So recently, single-cell RNA sequencing has been developed to significantly increase cellular resolution by barcoding single cells. So now we can identify gene expression and epigenetics signature of cell populations in tissues. Single-cell RNA sequencing has so many applications in neuroscience. It's widely used to understand cytoplasmic RNA and nuclear RNA, to understand DNA methylation and chromatin accessible sites, and more recently, to understand the gene expression in specific locations. This is very useful to understand the receptor and ligand relationships at tissue levels. Our projects, we propose that beta-cellulin or BTC is a novel therapeutic target for schizophrenia. Our data from postmortem brain tissue show that BTC is significantly lower in schizophrenia compared to healthy control and other mood disorders. And we next ask some questions. So what are the altered signaling pathways as a result of BTC dysregulation, and what cell population plays a major role in this pathological condition? And the second one is that can signaling pathways in BTC knockout mice be compared with the signaling pathways seen in schizophrenia patients with BTC dysregulation? And what we are doing right now is to perform the single-cell nuclear RNA sequencing in human postmortem brain tissues from schizophrenia patients. We aim to understand the transcriptome and epigenome, and also the spatial RNA sequencing in schizophrenia patients. And the second one is that we have set up a mice model for BTC knockout, and then we plan to perform the behavioral tests and also single-cell RNA sequencing in mice. Yeah. Yes.
Great, thanks Witaya, that was fantastic. Our next speaker is Ariel Dunn, and she is another postdoc in our lab that is working on the ARX project. She's going to give us an overview.
Thanks, Rachel. I'll just share my screen. Can everyone see that okay? Yep. So, hi, my name's Ariel. I'm a postdoc in the Behavioural Neuroscience Lab, and my project that I'm working on at the moment is looking at gene mutations and how they relate to psychiatric illnesses, with a particular focus on neural dysfunction, neural circuitry dysfunction, or electrophysiology changes. So as Rachel had mentioned with the RAX gene, which was found in a patient with schizophrenia in one of our human clinical trials. It's the Aristoteles-related homeobox gene, which produces the ARX protein, which is used in the brain for the migration of neurons and the communication between neurons, particularly in a specific type of neuron that is very important for the communication across the brain. And the ARX mutations that we have previously been looked at in the literature in males, it's very highly studied and has been linked to severe epilepsy and intellectual disability. But interestingly, the ARX mutation that we found was in a female human, a woman, and it's unstudied until now, but it has been linked to psychiatric phenotypes such as schizophrenia, or at least an increased risk of schizophrenia. It's located on the X chromosome and therefore is sex-linked. So this sex difference between male and female phenotypes is not really surprising. So a specific question that we are asking is, do the mutations that we see on the ARX gene convey potentially a sex-specific risk of psychiatric illness in females? One particular focus is on electrophysiology. So Witaya spoke lovely about the cellular level of exploration that we do in our lab, as well as touching on the behaviour, which I also have an interest in. But electrophysiology is the electrical signalling between cells and the surrounding tissues, and it encompasses small localised circuits of cells that communicate between one another, which also link to other circuits of cells, which then encompasses the whole brain and the regional looking at activity and the communication between neurons. So you could think of electrophysiology as the intermediary between molecular or cellular changes occurring in these models and in psychiatry and altered behaviour. And we can do this in animal models. So one of the slides that Rachel showed the little mice with the electrophysiology helmets on. This is what the rat looks like. So we can looking at electrical activity in humans non-invasively via electroencephalogram, such as the first picture, an EEG cap. We can do the same technique in animals to measure the same activity at multiple levels, including single cell during these circuits and the cumulative activity that you would see in an EEG. And these altered electrical signals have been associated with impaired cognition, psychosis, and other clinical symptoms relevant to mental disorders, such as treatment compliance and risk of hospitalisation. So two key examples of these electrophysiological markers that we might be looking at in humans and in the animal models is one, gamma oscillations, which is basically brainwave activity. The higher frequency a brainwave occurs at, the more communication and energy is being transported through the brain. So gamma activity, this bottom wave down the bottom, it's very high frequency and would be associated with someone with a very alert or a focused state of mind. So during a cognitive task or paying attention very closely, as opposed to say a beta wave or theta, which isn't shown here, which is a slower wave, which you might see if the person is bored or if they're sleeping even. So when looking at gamma oscillations, we can also look in the right-hand side of this figure, which is showing a heat map with the hotter colours showing more activation of brain regions and the colder colours showing less activation. This left figure shows a control healthy human, and on the right-hand side, we see less activation at the higher frequencies or the gamma oscillations on the right, and this is a schizophrenia patient. And we can measure these things in rodent models as well to try and determine what's happening underneath the electrophysiology to perhaps give us an idea of what we can treat potentially in humans. Another example of what we might look at is an electrophysiological marker called mismatch negativity, or MMN for shorter, which is basically an encoding signal in the brain for prediction of a sensory environment. So the blue line here is elicited when a normal, predictable sound environment occurs. So an example would be driving through traffic. And the deviant or red signal here, which gives a lot higher response, would occur when something breaks that prediction. So say a car horn in the middle of the traffic. And the difference between these two waves shows us an indication of how well our brains can predict changes in the auditory or a sensory environment. And in schizophrenia, this is robustly reduced and is one of the most robustly and widely reduced EEG markers that we have available to us. And funnily enough, we can measure this in the animals and modulate it in the same way that we can't in humans. We can modulate them by not only recording, but using optogenetic techniques as well as recording during behaviour to see how they modulate, and that activity modulates behavioural outcomes in the rodent models. So they're the two sort of focuses that I would be focusing on with the ARX project.
Ariel. Ju is next, but I don't think we have any students left. Jin, do we have any students online?
Sorry, it looks like there isn't any at the moment now. There were two. Yeah, two.
Just be presenting to us.
Yeah, I'm sorry. I don't know what's happening. Like, so we had about 130 registrations, and there was some, I'm pretty sure, that had mental health down, but they've been advertised through the faculty because we're not allowed to email people.
Yeah.
Would it be useful presenting anyway? Because I can see that it's recording. Maybe if, I don't know, people...
Wait, did you finish your recording? Sorry, did you you finished it, right?
I'm not sure. On my screen, it's saying that the presentation.
Yeah, it's automatically on record. Gabby, who organized the recording, is recording. So did you finish your presentation, Tori?
Oh, Ju.
Yeah. Sorry,
yeah.
Mine is, yeah.
Yep. So it's good. We can probably edit this bit out before we put it in.
Ju, did you want to do your presentation?
Is anyone want to do a project with me? Left here, Rachel, Ariel, Jim.
Look, I'm always down.
I mean, you know, I was semi sort of enthused because I got enough on my plate already anyway. So if I don't have to present, I'm happy not to. But if I should do it for the sake of the entire recording, I can do that as well. Yeah, I can do something.
Yeah, maybe you should present because it's recording.
Yep.
Just edit this bit out.
Sounds good. Okay. So I'll just share my screen. I'll keep it reasonably quick. I screw up though when I'm doing share screen, and then that's the one. Share. And then ready to record now. Okay. Yes. Hi, I'm Ju. So I'm head of the sex chromosome neurobiology laboratory in the Department of Psychiatry. So really the main premise, I guess, the research of our lab revolves around the inherent sex differences that are observed between the male and the female brain in the healthy or physiological conditions, as well as the differing susceptibility to disorders like Parkinson's disease, dementia, autism, ADHD, and the list goes on. So really the recent, I guess, the recent initiative by organizations like NIH and CIHR and European Research Group really are highlighting the importance of considering sex as a variable in biomedical research. So better understanding maybe some of the biological factors that underlie sex differences, or these brain sex differences may hold clues to uncovering new sex-specific causes of these neurological diseases, as well as potentially finding novel treatment options that are optimal for each sex. So really the research around the past 30 odd years has focused around the influence of sex hormones, and that certainly is well established because certainly the protective effects of estrogen in females in disorders like schizophrenia and Parkinson's disease are well established. But what we are really sort of interested in is the role of sex chromosome genes, such as the ones on the X chromosomes. And really the work that we've done over the past 5 years has revolved around this gene called SRY, which is a sex terminating gene on the Y chromosome germ. And it's involved in switching on the male sex during the embryonic development. But what we found is that SRY seems to be involved in function beyond, I guess, the initial male testis development. And what we're finding is SRY directly exerts male-specific actions in the brain that has a significant influence on dopamine-related functions, and dopamine is an important chemical that plays a role in a number of physiological processes like reward and addiction, voluntary movement, executive decision, and so forth. And what we find that SRY actually regulates the levels of these transmitters in the brain. And what we're also finding is that this regulation of SRY may underlie the disorders that are involved in dopamine dysfunction, like Parkinson's disease, as well as ADHD. So the principal goals of our research is to understand how sex chromosome genes influence brain function and behavior, to understand why males and females are differentially vulnerable to neurodegenerative and neurodevelopmental disorders such as Parkinson's, ADHD, and autism, and to potentially identify sex-specific risk or protective factors to develop more effective therapies for each sex. So the work, I guess, that we do really revolves around a variety of experimental approaches using animal models of neurodegenerative and neurodevelopmental disorders. We use a variety of tools to manipulate these sex chromosome genes. We then take, I guess, some of the findings that we see in the preclinical models into the human postmortem sections to see whether these findings are clinically relevant. And so we are adapting, I guess, much more sort of, cutting edge tools such as human iPSC models to see whether we can identify novel drug targets using a clinically relevant model. So if you're interested in the research, you can get in touch via my email, or you can certainly look at my details on the Monash Psych website. Thank you for your attention.